So it's my great honor to introduce Professor Wei Wang from UCLA, where she is a professor of computer science and also a director of Scalable Analytical Institute at the UCLA. So her main research interest is in big data, database systems, and data mining, and also bioinformatics, cover many different areas. So he has won a number of awards, so I will not list them. I just want to mention that he won this NSF Career Award in 2005 and also Okawa Foundation Research Award recently in 2013. She's very active in this community. She served in a number of editorial board and program committees in a number of conferences in data mining and database. So without further ado, we welcome to UCI.
Thank you so much. Thank you very much for giving me the opportunity to share some of the current work at UCLA. So,
Micro.
Is this better now? Thank you for reminding me. Oh, that's a feedback. So, again, thank you for inviting me to come over, and also I have my colleagues on the front row, so really appreciate they also come. Actually I joined UCLA about 2 years ago. I graduated from UCLA a long time ago. I don't want to mention the year so that makes me look so old. Today I'm going to share with what's actually only 2 projects was happening at UCLA, and hope to get your interest. So nowadays data is everywhere. I think for this crowd we don't need to say like how important the data are, because we all realize from computer science that's especially from the field of database, data mining, knowledge management, and big data, we all appreciate the volume, the diversity, and the challenge that's posed by those big data nowadays. So what happened at UCLA is that we recently started the Scalable Analytics Institute, started with 4 core members, showing the picture here. So actually one of them, Tyson, is there, so he's really a star. So this is actually the big picture that for the institute want to achieve over the long term, and that's why we have a collaboration, ongoing collaboration here, thanks to Tyson. Actually, he's really the key person here. So to basically provide a unified system that to not only be able to support the batch, interactive, and streaming mode, but also especially design and the next generation framework that actually can efficiently support many of the machine learning data mining tasks. So because my background was data mining, I was trained as in the data mining field before I started to do other area. So I always approach a problem from a data mining perspective, and you will see that from the project that I'm going to share with you. So in our center, we have many ongoing projects. This is just a very high level, a short list. There's many more that has actually not listed here. So and we are also looking forward to start new collaborations. And today, because I know I have an hour, I'm going to share with you some of projects that I did by me and my student, like actually on the machine learning end rather than on the system end of the work. So I started to do bioinformatics, actually start to get exposed to the field of bioinformatics and computational biology when I joined University of North Carolina about 12 years ago. So before then, I was only doing pure data mining, nothing else. It was a fascinating field. What got me most interested, not just the big vision they put forward then, but also the huge amount of data they were collecting then. Well, nowadays, when we look back, like 12 years ago, those data were actually small data compared to the data we have nowadays. But nevertheless, I was fascinated by the challenge that they put forward, and then I think, oh, maybe as a data mining scientist, I can do something to help. I hope to do something to help. So just to share with you what thing. Actually, back then this movie was not that old. So, but it's a good movie. So it's just a clip, and then.
Neurological condition, 60% probability. Manic depression, 42% probability. Attention deficit disorder, 89% probability. Heart disorder, 99% probability. Early fatal potential. Life expectancy, 30.2 years. The name for the certificate?
Okay, so at least I think I apologize for this this problem, but I can see that at least people sitting in the front row may hear that the punchline here is that you have all these probabilities, right? From derived from just a single blood of a single drop of blood, right? So that's what actually how Hollywood envisioned that was going to be the future of our society and the medical field. But what the reality was that, of course, we have not reached that stage yet. We're working towards that, but this is actually a good long-term goal that we want to achieve, right? Because not like in this movie, well, once we decide that this kid doesn't have a good probability to survive, and then maybe they don't put in resources right there. But on the opposite, actually, nowadays what we're trying to do is that if we have this information, because there's quite a few diseases, so actually we can put in an early intervention to actually delay this process, or maybe even completely avoid this bad outcome. So that's the goal. But what's complicated in this process is that all the disease and how they started, how they form, how they progress, is not as simple as we hope for, right? And things are complicated, especially that it probably doesn't, to our surprise, that genes, well, from our biology, high school biology, and we have more than one genes, right? And then they do not work alone. So it's expected that actually interact in some way and play function together. Well, so this is the still ongoing research that in order to understand their function, how they performs. But what we know by far is that for many diseases, actually more than one gene play some role in it. Okay, and what we discovered, this is a number of years ago, we used mice to study colon cancer, and these two genes on different two chromosomes actually. Both of them are playing very important role to the growth of the tumor. But what's create a difficulty is that if you examine the effect of one gene at a time, maybe you knock down the expression of the gene and see how that works. So actually you do not see a very strong effect. But only when you see both of them together, examine them together, then you start to see, wow, actually the effect is not additive, right? It's far more than that. So you can see that this creates complications because since we have, maybe humans have like 20,000 genes, and then we don't know which subset, what combination, right, to start with. And obviously we cannot afford to examine all possible combinations. Like coming from a computer science viewpoint, this is a combinatory search problem, right? But unfortunately, we cannot do this search in the medicine field like we did in the computer, right? So that we can design an algorithm to do search, but each of those experiments take money and time, a lot of resources, so that they cannot really afford to do exhaustive search. So what we said, well, then how do we, as a computer scientist, help with that? And so the signals may be in the data in somewhere, right? But we just need to be able to find out, to derive or generate the right hypothesis for them to test. Because if we tell them this combination, then they can design some experiment to really validate that. But if I don't tell them, I say, well, try out pairs, then they don't know what to do. They probably wouldn't do anything because that would be too expensive for them to do. So then we start to say, well, how do we help? right? So we realize that to solve this problem, we have to consider at least two aspects. One is from the statistical viewpoint, right? How do we capture this interaction? What do we need to do, right? What kind of tests do we need to do? And then we actually, for this, we consult a lot of statisticians and biostatisticians, and then each of them actually give us a slightly different recommendation, right? They always have their favorite test. So, and then the second is actually where our expertise lies is really from the computational side. There's hundreds of billions of potential interactions that one could test. And how do we fish out, right? Maybe rank them in certain ways that so that the biologist or the doctor can use that to guide their experiment to validate all those interactions. So how do we actually efficiently compute those tests? And so there works in in the past 10 years, starting with some kind of exhaustive search from data. So basically, they really try all possible combinations they could try, look at the data, and run statistical tests to the interesting trait, that the trait they're interested in. But you can see that this approach is not scalable, right? You cannot afford to test many of those genetic variants that we observe in the human or any organism. And then people start to say, well, how about we use some heuristics? And then one heuristic commonly used is that actually it's a two-step approach. The first step is that they check each individual variant and one at a time. Do a screening. It's a linear time screening. And then they set a threshold. Okay, only when the individual, the marginal effect is above some threshold, then I leave them aside. And then in my second phase, I try out possible combinations of those ones that pass my threshold. You can see that, well, that's an interesting approach, and it worked, it can help to find some interesting interaction, but it has a limitation that if the marginal effect, the main effect is weak, like the example I showed you in the past, then you probably would never try the combination because it's never past the first phase, right? So that would actually make you to lose maybe some important interaction that's crucial to the disease. So, which means that there's still more algorithm development that helps them to fish out the right information. So we want to approach this from two angles. The one is that we want to make this computation very efficiently, right? We cannot really speed up the real biological test and validation, but at least from the computation viewpoint, maybe we can tell them, okay, once you give me the data of your genetic variant, then I can tell you which hypothesis to test maybe in a couple of days. So that they can accept. But if you say, well, give me the data and then see you in 10 years, because that's how long my computation is going to take, then that will never happen, right? So we want to improve the efficiency dramatically because we did the back of envelope calculation. If we use any brute force way to implement that, it does take 7 to 10 years per data set. So, and the second is that we don't want to compromise the quality of the outcome. We don't want to give you the wrong hypothesis. We want to guarantee that the result is optimal in the sense that it would give you exactly identical result as if you have the infinite amount of resources. So that's our goal. But we want to rework the computation so that it will arrive at that result much faster, okay? And then we also want this approach to be generic, that not just limited to one specific statistical test, because it seems that every statistician have their own favorite test, so that we want to be able to support most of them, if not all. So since this is a computer science crowd, and I want to have a biology 101, that information that are necessary to understand the algorithm. So from our viewpoint, you can see that I have 2 mice, and then each of them has their genome sequence. Okay, I know the genome sequence is far more complicated than I write down there, but for now, let's assume each is a single sequence. Okay, actually it's not single sequence, but let's assume that. And then you can see that for most of the sites, I align them, and then they are identical, right? And then, but there's a few places, well, actually one places on my chart, they differ, right? This called a single nucleotide polymorphism. Actually, it's most common variant that we observe, okay? And those are interesting because if these two mice exhibit any difference in their phenotype and the trait, like in this case, the coat color is different, right? You would imagine that probably caused by one or more of those variants, right? But we don't know which one, right? So, and we want to identify which one has a statistically strongly correlated with that. So, and then what we have actually is not just one of such variants. We have many of them. Actually, there's millions of those. Okay, already be identified. And now we have a set of samples, and then we have their genome sequence, and then I highlighted those SNPs. We call it SNPs. And then you can see that because we're interested in that, like which SNP or which combination of them may cause the difference in the phenotype, then I'm just only going to focus on those. And then in this case, I actually encode them in a binary variable. The reason here is that because they are typically caused by a single mutation, which means you flip from one base to another. So you only observe two variants, even though there's 4 possible variants. So that's why you can encode them nicely as a binary matrix. But you know that this is a very simple case, and probably the simplest scenario, because in human it's more complicated than that. But we have extension to handle that if it's not binary. But for now, let's assume it's binary. And then we have these millions of those SNPs. You can see this genetic variant is represented as a huge binary matrix. And then that's what we work with as one piece of the data. Okay? But then on the other side, we're interested in all those phenotype variants, right? At the very beginning, I showed you an example of this colon cancer, the tumor size. That's one phenotype. And then there's maybe other phenotypes, so that there's just some examples like coat color, body weight. You can see that it really exhibits a huge difference, a variance, right? And so that's actually those are laboratory mice, and then that one is actually used to use as pet, so the white one. So it's really like a cotton ball in your hand. So. Well, I didn't put it in my hand, in somebody's hand, I saw that. So, so now the question is that I have these binary panels, and I have this phenotype. Let's say this is maybe the body weight, okay? It can be anything. And then it can be either categorical or numerical, right? And then my question is here, well, which SNPs, or combination of SNPs, that together actually cause this variance? So basically, we want to do a statistical test. You can see that if the data size is small, that's trivial, right? That's problem being solved a long time ago. So there are standard solutions, and the question is that which statistical test you use, and then you can just follow what the guidance offered by the statistician. But then the question is that, well, we may say, well, we only have one phenotype of interest, but on this side, the genetic variance is like, think of you have millions of those, at those columns. So, and then what typically people do is that in the past, they basically doing a sequential scan on this side, right? Doing one column, because each column is a SNP, right? At a time to run the test. Okay? And then they have this test score there. And then they set some kind of a threshold. Okay, if it's past my threshold, I will believe that's significant. And then I'm just going to output that. I can even rank order them by the value, right? So I can do that. But then, well, how about combinations of them, right? Because that's where the interaction comes into play, as I said. So that, let's see if we just interested in pairs, only 2, right? That's the simplest interaction that we could imagine. Then you can see, well, that becomes, you try to test all the pairs, right? So basically that's already the quadratic number of a test that you need to do. And then the problem is that because you have you said, well, now we have many tests that we need to, not just a single test, right? And then how do we define this significant associated, right? Because people think, wow, the simplest way is like I just have a threshold, or I just return the top K. But the real scenario is not as simple as that. One problem is this multiple testing problem, because once you have more than one test, right, as you increase the number of your tests, actually the probability of seeing at least one false positive quickly approaching one. So basically you're guaranteed, if you're doing millions of tests, you're guaranteed to see a few, because if a few of them showing as significant, that's probably all like, sort of quote unquote expected because they're false positives. So we have to control that. Well, if we talk to statistician, one of the suggestions we may get is that, well, let's run permutation tests. Let's see how do we run permutation tests to help to eliminate this situation. So what we do is that instead of just using this original phenotype, we reshuffle the value, okay? Then by reshuffling, basically we break all the associations between this left side and this, right? There shouldn't be anything significant after we randomly reshuffle this. And then we do that. We do that multiple times. Each time we record the best score we can get, the most significant score. And then do it again, then do it again. And then let's say we do 1,000 times. And then we already sorted from the most significant to least significant. And then we said, well, let's take the third most significant one as a threshold. And then if the score I get from my real phenotype without shuffling is better than this, then I believe it. If it's not, then I think that's still false positives. So, well, that's fine if your data set is small. But the question is that we already have this second step, right? We already need to do many millions of pairs of those tests, but suddenly, in order to establish the threshold by permutation test, we have to multiply whatever the amount of time we spend by, let's say, 1,000 or 10,000. That's the number of permutation tests we need to do. Suddenly, that the computational burden is, is multiplied by, many times. So basically, this is very intensive, and we really cannot, even with the modern computer, even if you run it on a big clusters, it still takes a significant amount of time, because when I started to do this when I was at UNC, and literally our job got killed. So because other people complain on the cluster, because we cannot run longer than like a week or something like that. So you can see that. And we don't even dare to tell them at that time how long we really needed. But this is any brute force implementation is actually not trackable. So then we say that, well, this is a revisit of really summary to the challenge. I said that first, we can rely on statisticians to tell us which test to use. But then on the other side, on the computational side, our task is how do we finish all the computation, all the tests? Because the number of tests, if we only have, let's see, for a moderate size data set, 500 case SNP. That's not all the SNPs, right? But that's typical numbers output by array, a microarray. And then let's see, we only use 1,000 permutations. Then the number of tests is 125 trillion, right? And then you can multiply that by how much time you're willing to spend on each test, right? And then that number can be easily large, because now that I'm going to show you later that we moving from microarray to sequencing, which means the number of variants that we can generate is a lot more than 500K. So you can see that this is actually create a lot of a big computational challenge. And we want to basically handle this all together. Because if we knew we're going to do 125 training tests, right, altogether, then probably we can think of ways to speed all of them up. Because ways we can come up with will be, well, maybe some of the computation can be reused between tests, right? Maybe there's some bound we can estimate to help us to guide us to look for, to go directly to the places where there's a chance there's some significant association can be discovered. So we explore all of them. Because I don't know how many of you actually are from data mining field and also are trained with data mining. Because a priori property is probably one of the famous property actually published, right? The data mining paper that tells us how do we supposed to if our metric satisfies this property, basically it says that well we can use this to help us to prune the search space. The idea is very simple. It's like well you count the frequency of the item set, right? Count the number of occurrence of a combination of items, and that frequency actually monotonically decreases as you insert additional item in the item set. So that you know if you start with singletons and then slowly add items to your set, and then you're guaranteed that they call the support, actually the frequency go down and until some level that go below your threshold, then you can stop. So you can guarantee the result is optimal in the sense that it give you all the frequent item set, but you don't need to search the actually the power set of all items. So that's the nice property that actually Rakesh originally proposed, and then from that point, many solutions provided in the data, actually proposed in the data mining field have that flavor. So basically, you want to have some kind of property to help you to decide which part you don't need to compute. Okay, but unfortunately, this doesn't hold for our problem because we cannot easily define that. Okay, one test result is. You can easily depend on the other test result. We investigate that. That's of course our first attempt, but that didn't work. So, but then we go to okay, how do we maybe look at the intermediate computation, not as final computation, right? And let's see if we can cache and reuse some of the computation to help. So, and then so the way we prune the space is well, we want to derive some bound, actually upper bound of test statistics, and to tell us whether some tests that need to be performed or not before we actually compute the test. The second is that we want to create an index, a simple, very simple index to help us with the bound estimation, to help us to guide us to do the research. And also we want to carefully design the order of computation in a way that we can maximally reuse the computation. So first, let's do the bound, and then let's look at with a specific test. The first attempt we tried was we call it fast ANOVA because we use ANOVA test. Because ANOVA test is probably one of the common tests that people use, right? And then we say, well, can we speed up? Let's assume ANOVA test is the one we use, but how can we speed it up? Okay, apply all those ideas that I just said. So how do we do that? The way we did is that I didn't show you the actual formula, but the idea is that if you want to test a pair of SNPs with a phenotype, okay, and then we can have an upper bound, we can rework the formula such that the upper bound is not strictly equal, it's an upper bound. It's a constant, which we can safely discard because it's a constant, plus two functions, R1 and R2. And in a way that they are a function, so R1 is function of NA. What's the NA? Is there's two columns or two SNPs that I'm interested, and then if the left on the X side take 0, and what's the minority count of this XJ? And then that's my NA. And the NB is when Xi takes 1, what's the minority count here? That's my NB. So it's very simple statistic to collect. So, but you can see that those are two parameters I have. And once I can decompose the upper bound into this way, so this only depends on NA, this only depends on NB. So, which means that if the actual SNP pair I'm interested in, right, because there may be multiple, many of those pairs that have the same value of NA and NB. That's possible, right? Because I only look at the minority count. I don't care which sample carry that count, right, carry that minority. So, if they share the same, they would have the same value, they would have the same upper bound. So that's give me a way to actually group and index those pairs so that we can quickly estimate the upper bound and we can quickly perform the test. So that's the idea. So in order to do this, I can see that I have this 2D array, right? NA by NB. So that's each entry represent, actually we use that to index the set of pairs that have the same value of NA and NB. So we do that. So we can see, you can see that we can go through the pairs, the X1, fix X1, and do X2, 3, 4, 5, and then we can insert them. So, but we can even optimize this process to make it really efficient. But the main idea is that we can do this exercise, and once we do this, we can see for a given entry, we actually have a quite long list of them share the same upper bound. And then we can use this upper bound to tell us whether any of them may have a chance to survive. Because if the upper bound is even lower than our significance threshold, then none of them would survive, right? We don't even need to compute their actual test score. So that gives us a lot of savings. So you can see that we compute this purely on the genetic variant matrix, the SNP matrix, has nothing to do with the phenotype, which means that the exactly same data structure can be reused when we test the original phenotype, and it can be reused when we test for each of the permutation, because we only permute the phenotype. We don't never change the original, the binary matrix we start with. So that also gives us a lot of sense. Essentially, each additional phenotype only use epsilon amount of time. So that can allow us to do it very efficiently. So what we end up doing, this whole process is very simple. So we basically first create the index, and then for each permutation, and then we basically use that use this index to estimate, to basically find what we call candidate pairs. That basically those ones with upper bound greater than the threshold. And then for those we compute the test value. Okay? And then it's a similarly very simple idea. And you can see this is a very small data set, so that we can run the brute force within reasonable amount of time to show you that actually this gives you 3 out of magnitude speed up right away, okay, by doing this. And so you can see I have the number of SNPs I'm interested in is only up to like 42K in this case. It's a very small data set to show you that. But even for larger data set, this level of saving in computation actually is consistent. And then we see that, well, because of my saving is coming from the pruning that I was able to do with the bound. So you can see that with the the error rate is like decrease. Actually, the amount of saving I'm able to do can further increase. But nevertheless, even with on the very left hand right side, it's over 99.7 percentage computation that I don't need to do. Okay, so that's give us a huge advantage in terms of the speed. Okay, so that was our first work. But since then we say, well, ANOVA test. What if they don't want to use ANOVA test, right? What if they prefer some other test? So we sort of start to look at other tests to see, well, can we do that? Well, it turned to be that almost all the tests that you can imagine can be decomposed in this way. So basically, it's constant, but it would be a different constant plus 2 functions. And then those 2 functions are actually, there's 2 separate input parameters that control this. So you can always apply this idea to other tests. And therefore, we generate actually we develop a general framework for this. So it's almost doesn't matter what test you want to do, but you do have to write a little plug-in for the specific for the test. But the general framework is actually can work well for. Actually, we even do further permutation for the general framework to achieve another 2 order of magnitude speed up. But I don't have time to show you that. But the idea here is that even similarly for this problem that actually was studied by biostatistician, by genetics for quite a long time. But when the data set grows to some level and then they face this hurdle of the computation, and then we as a data mining scientist, we can come in to help, right? To apply all these data mining skills to help them to design a better way of doing the computation. Because whenever I present to a geneticist, and then the first question I was received always like, what do you miss, right? Because you show me that all this impressive speed up that you must compromise something, right? So I have to show them, like, that's why I start with that. I showed them like there's a little data set. He's saying that, well, we really can prove we get you exactly identical result. And then if you're happy with the old way of doing that, using that test, and then But nowadays, because of the amount of data, you cannot no longer do that. But then you can use this to get exactly the same result as you did before. I can prove that. So they're happy with that part. So this one was used quite widely. So the next thing is that, well, during the past several years, and the people moving from doing like a micro type of experiment, and then to actually high-throughput sequencing. So basically, you can see this is I borrowed this chart from a paper so that you can see there's a lot of different applications that sequencing can help. So what is it, right? From a computer science viewpoint, and so how does that actually related to what I did before is that let me show you a simple pipeline. So you start with some samples, right? That's people, patient, and then you extract their either DNA or RNA depending on your task, and then you fragment them, because nowadays the current technique they cannot really take a molecule or the DNAs, read from the first base and all the way to the end. And then because the quality of the reading drops dramatically after you pass maybe 100, right? Because they shine the laser beam to the material, so that is really break down deteriorate the sample and then impair the quality. So basically what we have is that we have all these fragments, many of them, like many million of them per sample. And then this sequencing machine basically read it out from one end to maybe up to 100 or 125 base pair. So basically we end up with a huge bag of short sequence. And then we don't know where they came from, because once we share them into the fragment, we lost the information like where they came from. And then you can see that actually, because everybody is adopting this technique, and then the amount of sequencing done is it's increased over time. This is a paper from a paper a couple years back, and then you can see that is actually the growth of the sequencing data already surpassed the increase, the improvement we can have on the computing side. So basically, this is computing is the hardware advances. You can see that which means that they really cannot wait for the computer to become faster, more powerful to solve their problem, because the gap is going to be only bigger and bigger. We have to think it from the software end, from the analysis end, to say how do we help with this. So typically how they did is that there's many different variations of this depend on your final task, but typically you take those short string, and then you align them back maybe to the genome. The reason is that you want to re-establish where they came from, right? And from this aligned file, and then you assemble, in this case, in this chart, it's actually all those, this is RNA sequencing. So basically these are the transcript, okay, to see which gene got expressed in which form, okay? So, but they have these 3 steps. And then there's algorithms developed for that, and then I only show you 3 of them, combinations, like alignment, and then the sampling step. And then that's how long it takes for one sample, one small sample in this case. Okay, so this is, you have to do the calculation. This is about like maybe 20 hours or something. And then this will be like a day or maybe more than a day, just one sample, okay? And then I think this is one sample even on 8 threads, so that do that. But you can see this is significant amount of time, but doable, right? People are doing it right now, right? So, and, but then, our thinking is that we start by doing that, and then I started at UNC, and our first batch of data had 200 samples, and then we start to run that on the clusters, and then again it's like it's a little bit time-consuming because we have to play with the parameter, because all of the algorithms have a number of parameters to adjust. But you can see that once you change parameter, then you have to restart your job and then see the result, and then you have to store the possible result, and then the result is huge too, and then so that didn't work out. You end up like, since none of my students is in the audience, I can safe to say that at that time I was frustrated in the sense that when I talked to my students, right, they said, oh, I'm waiting for the result. And then that's another couple of days it elapsed, and then once the result comes back, well, not quite as what we expected. Let's change the parameter. Because what are the parameters? Because when they do the sequence alignment, they basically what's the amount of error you'll tolerate. That's one parameter to control, right? So, but once you twist that a little bit, then guess what? Another week or 2 weeks just pass just to get that result. So I was quite frustrated. Another that time we were part of NIH Center for Excellence, so we have a site visit coming in a month, so that we can't really afford that. So I told my students, like, okay, you can let the machine to compute, but then you cannot be idle, right? You have to think about ways to speed this up. So then we said, well, how can we do this? And then there's other things, because other artifacts that once you start to alignment, and then typically those algorithm to trust the result of alignment, thinking that if I can align, that must be the right location that this read came from. But it tend to be not as simple because the genome is full of repetitive sequence. Really confuse alignment algorithm. Sometimes you can align to multiple places. Sometimes even if you can align to a single places, it may not be the right places. Okay? So, and then that's what would cause like subsequent analysis to give you the wrong result. And then that's what we experienced because it turned out to be our very first result. We want to see between two groups what are the genes that express differentially, right? Then it turned out to be all the results are either from my collaborators' viewpoint is like either we made the biggest throughput in science over the past 2 decades because we discovered so many things that contradict whatever the belief, right? Or there's something wrong with the result. So of course, what happens is there's something wrong with the result. It's actually because it's not our own software, so we took the recommendation from the sequencing company. So it's one of the most widely used result, but people just use it without realize the artifact in the result. So it turned out to be all those results are false positives. There's none of them are full positives. So that's make us think we have to really rework the algorithm to basically overcome the problem. And then we start to look into what are essentially those algorithms does, right? So basically we know that they want to align the read back to the genome. And then like this is an alignment. And what they do is that once they align this, they look at this pile up distribution and then estimate the abundance. So basically this is the curve, and essentially what they estimate is the area under the curve, okay? In some sense. Well, there's normalization, there's a things bias correction, but essentially area under the curve. And then this is area under the curve, and those are the popular algorithms that people use. And then to basically take this alignment as the input and estimate area under the curve. But of course, if the alignment is wrong, then their estimate will be way off. So I'm starting thinking, well, can I do this without alignment? Because when the alignment is wrong, it's very hard to crack that unless I start to realign, because I don't know the ground truth. I don't even know where I should realign. So the approach we're taking is that, well, can we do this without alignment? Because our final task is not alignment. It's rather to estimate the expression level, right? So, and then, of course, we want to speed it up, right? Because I don't want to spend time there drinking coffee and waiting and chatting, and I can't even blame them, right? So, so they did use that spare time to come up with some approach. So, and then our approach is that, okay, let's try to, we call it either estimate or approximate this size of this area, but not by really specifically to sort of compute or what is the height of this curve at every single position. Because what we observe is that actually there's a strong correlation between those neighboring positions in terms of the height of that curve. The curve, in other words, this is a rather smooth curve, right? If you really zoom in, it's a rather smooth curve. So our first attempt is okay, let's define some feature because it's a data mining field, right? People from data mining field or machine learning field that we are familiar with the term feature. And then our feature we call is a k-mer. So actually k-mer is a string of length k. It's widely used in many of the bioinformatics algorithm, or in the old days in computer science, in the string finding like substring, indexing substrings. So it's a very simple feature, but there's many of them. Because the k cannot be too small, because otherwise it's not unique, right? So our k is between 40 and 60. So you can see there's a huge number of all possible k-mers. So it's become a feature selection task, right? So we wanted to select the smallest possible set of k-mers, such as they can help us to accurately estimate this area in the curve. So that's our goal. So first I said that we probably don't need the feature to be, if the feature is a k-mer location, to be like always adjacent to each other, because there's a strong correlation between their reading. So that we want to basically maybe just sample some places, right? So as long as those places give us accurate reading, right? Because as I said, there's repetitive sequence, there's noise, there's errors. Not every position are equally informative in that sense. Some positions maybe are more prone to those errors, and then some are more reliable. So we want to pick those more reliable, we favor those more reliable ones. And as long as we select that, we can use those basically the count of those k-mers, right? Then to help us to estimate that. That's a high-level idea. But of course, it's not as simple as I said. If we can achieve that, we may not need alignment anymore, because alignment takes most of the time in this exercise. Okay, so the challenge here is that multiple transcripts, multiple of those, may share some common or very similar subsequence, right? That's what we know before, because otherwise we wouldn't have a multiple alignment or confused or wrong alignment if everything is so unique and distinct. So we have to be able to address that. So the way to address here is that we take all the possible transcripts. We run a clustering algorithm on them. Basically, essentially, we want to group all those similar ones together, so that we can sort of like look at them all together and then do our inference, right? So basically, because we want to use k-mer as our feature, so our similarity measure for clustering is basically how many k-mers they share. Okay. Then we can take any clustering algorithm to do this exercise, and then once with that, we can see we want to find all those k-mers that actually are unique to one cluster but not appear at all in the rest of the clusters. And we call them sigma because they are signatures of a cluster, right? And it turns out to be there are many, many of those once we do this. If we don't do clustering, we want to sort of, for each transcript we do this, and then there's not many there that are actually a signature of a single transcript. But once we start to cluster them, because the similarity between clusters is low, so we can easily find those ones unique to one cluster but not the other. In this case, in this example, you can see that my k is 4 right now, so in practice it's probably 40. So you can see those GGAA is unique to class 1 but not in class 2. And so is GBTT and the TATC. And in class 2, you have ATTT. And the ATCG only appear here but not there, right? So we start with this collection set of sigmas, but we don't need all of them because there's still quite a lot of them, right? We still want to select the more reliable ones from them. So we have this, and then we first find out the sigmas, and then we see, well, now we have those the read, right, or the bag of short sequences, and then we can count how many times each of these appears. Okay, how many times each of these appears? So knowing that this is not doing alignment, so basically just a k-mer search, right? Many of those k-mer search, and then we can basically have all this k-mer listed, right? And then from that, so essentially, basically, we can treat this this k-mer that we use as a special type of read, right? It's shorter than the original read, but that's more reliable than the original read to give us the estimate. So it's kind of, because we don't use all of them, it's actually a sampling. We're doing like a feature selection. And then we can sort of like safely discard the rest part of the transcript database, and then we don't need to look at them because we only use the k-mer to do this. So what we do is that, well, essentially we want to estimate how much each of the the transcript, is expressed. We have this variable alpha, right, one associated with each of them to tell us how much in the percentage ones, right? It's a 2% at the top, and then 3% is alpha 2, or so on and so on. We want to infer, that's a known variable we want to infer, right? So what we can do is that it's a maximal likelihood estimation, and then, but we can do this within each cluster because all those selected k-mer, the sigma, are not shared, right? Because they are unique to a cluster, so there's no interference between clusters in terms of the reference and inference. So we can basically essentially decompose in one big system into many, many small ones. So we can parallelize them, we can do all kinds of things to speed it up. So as opposed to the alignment-based method, well, in their very last step, they also do maximal likelihood estimation. It's actually, from a machine learning viewpoint, they are quite similar. The one difference is that this is a one big system to optimize because they have everything there. And we have several thousand, typically, small system to solve. And it turns to be much faster. And now here's show you the overall picture of this process. And as I said, we have this clustering first, and then detect all the sigmas in the raw read data, and then doing this quantification maximum likelihood assessment. So there's two steps that we actually engineer some of the optimization speed up. The first one is on the finding all the k-mers, okay, all the sigmas from this. So by definition, we say that this k-mer only occur in one cluster, and we can have a straightforward implementation. Basically, we can just count, right? So for each of the k-mer appear in one cluster, whether it's appear in another or not. If it does, we throw it away. Otherwise, we keep it, right? But then to store all the k-mers, we need a lot of memory, right? And it's not like commodity computer, we can have that big memory. So we use Bloom filters to basically, because Bloom filter is good for like, if you have a set of elements, right? You want to test whether one given element is in the set or not, right? So it can quickly test that it's a probabilistic because it's actually using a bunch of hash functions and to do it in a probabilistic sense. So by doing that, we don't need to store the actual k-mer anymore, right? We can just use a few Bloom filter for this. So even though maybe there's one sigma, one k-mer that satisfies all the criteria, but we miss it, right? But since we don't need to use all of them, right? We in the end, we only select the subset anyway, so it doesn't matter if we miss some, right? But we need to guarantee that the one we selected actually does not appear in other cluster. That part we need to guarantee. And the Bloom filter does perfectly in that regard, and it's very fast. And then the next optimization is really, well, once we have this, we want to basically detect all the sigmas in the raw read data and count how many times each one occurs, right? So in this case, we actually, again, brute force is k times n. N is the size of your read, and then that's typically quite big, and then k is the length of your k-mer, right? And then to just speed it up, we use this rolling hash algorithm. So basically, speed up from order of kn to order of n. Idea is simple, and then so basically you have this read, and that's your k-mer length, and then basically you do this sliding window, right? And then every time you only update your hash function in constant time because of the k-1 overlap there. So of course, that requires special design of the hash function that have this property that you can just reuse part of the computation. So that's how it becomes linear time. So after we did that, well, we were happy with the speed, right? As it's faster, but we don't know the quality. We want to demonstrate the quality of the result is not compromised. And since for the real data, we don't know the ground truth, but we compare with the experiment run on the exactly same samples, but with older the microarray technology. So we compute the correlation. So these are different. The first one is our method called RNAseq, and then they are competitors, right? Those three I show you, the running time is very slow, and the Sailfish is one of the recent one actually published this summer. They don't use alignment either, but they use all-kamers. They still try to solve a big system using all-kamers. But this is independent because we also published this summer, so these are their independent development. You can see that no matter we use Pearson correlation or rank correlation, the results are actually, all of them are quite similar, right? They have a same level of correlation to the old macquarie result. The macquarie is not the ground truth either, it's just an independent measurement, right? So ideally, we want the correlation to be 1. We know that, okay, if they're independently measured and they're completely correlated, right? So then we know that we both get it right. But around 90% is not too bad. So, but when we look at this running time, the CPU time, so you can see that this is on one sample with 44 million of pairing read, and then pairing means you have 2, right? You read from both ends. So, and then you can see this array scheme takes about 10 minutes, right? And where the rest take about hours or days. So that's actually that's what I forced my student did. So in the end, actually, it's quite useful because it used to be like we get some data and then we want to get the result, we have to wait for days. And then because we often need to meet with our collaborator, and then that's really delayed the progress because we have to schedule a meeting, but with 10 minutes, so basically we can really get it run where we're in the meeting. And then basically I'll maybe go to grab a cup of coffee and then come back, the result is there. So that becomes very efficient. And then actually we even show that in the simulated data in our paper that the result actually quality is actually moderately better than all the previous one. But we don't want to claim that because we don't have a lot of results to demonstrate because it's only based on simulated data and then based on one set of experiment we did. So we just basically claim that we deliver comparable results but with much faster speed. And also, our implementation is only a single thread implementation. So we think because the way we're doing that is so easy to make it parallel. So, and then that's one advantage, but we have not really have time to get that done yet. So there's a lot of room to improve further. So this is about to conclude my talk. These are the references if you are interested. And this is actually, a sample, an image of like everything else that actually we did in the past. And so not intended to let you to read the tiny font, but if you're interested, we can talk about specific areas of research. So thank you very much.
So we have time for a few questions.
Can you go to the result slide? Your comparison with Express and RNAseq. The next one, the comparison of the CPU time.
CPU time, yes.
Yes. I remember the Express is implemented using an online EM algorithm, which in their paper they claim they are faster than traditional batch EM algorithm like couplings and RNAseq.
Yeah.
So I think at least Express should be faster than RNAseq and couplings. I don't know why. It's slower even than those two. So
that's, it's an interesting point you bring up. We read the paper, we also don't know why that's the case. But we ran, I show you this is for one sample, but we have like 200 samples, right? It's always slower.
Okay.
It's always slower. So, and I don't want to say that they make full claim, but
okay, yeah.
But we already sent an email to them a few months back to tell them that unless maybe we did something wrong, so that or maybe the system configuration, the requirement will be different. So then then we don't experiment the same same speed. So yeah.
So I have a question regarding the first part of your talk. The idea of fast novel is very clever, smart, and the results are very impressive. So here you assume that the genotypes of each SNPs is either 0 or 1 binary.
Yes. Yes.
Can it be used for 0, 1, 2?
Yes, so that's a good question. So the part I didn't show is that in our generic framework, actually, we can handle heterozygosity. So it's really 0 and 1 and 2. And then we also can handle as, because in the fast ANOVA, actually, the complexity still depends on the sample size. But in our generic framework, we actually further optimize that. It doesn't matter how many samples you have. You can run some thousands of samples. We actually run this on a big study for schizophrenia. It's about 5,000 patients over maybe 600K SNPs. And with EQTL, we have the gene expression as a phenotype. So that can work out. Yeah.
All right, so let's thank our speaker again.